All right, so the topic that we're presenting today is DNA cloning, and the presenters are Thelonious McDade, Eric Solis, and Corbin Greer. Okay, so this presentation is 28 slides long. I will be presenting the introduction slide and then slides 2 to 10, which go through the history of DNA cloning. Slides 11 to 18 will be presented by Eric Solis, and these slides will explain the science behind DNA cloning and how it works. And then slides 19 to 24 will be presented by Corbin Greer, and these slides will explain the relevance of DNA cloning and the recent application of the DNA cloning process. And then slides 25 to 28 are the references. All right, first I want to explain what DNA cloning is. DNA cloning, in simple terms, is taking a piece of DNA and putting it in some sort of biological carrier that will copy that strand of DNA for you. A lot of the time, people get confused by the difference between animal and DNA cloning. The difference between the two is that animal cloning only, or DNA cloning only copies a specific region of a portion of an organism, and animal cloning copies the entire organism. DNA cloning is done for a couple reasons. The first is to study gene structure, the second is to study how a gene is regulated, third is to produce proteins, and the fourth is to build different systems with different pieces of DNA that are strung together. One of the best examples of DNA cloning is the production of insulin, which helps in the medical field. All right, so the most important step to be able to perform DNA cloning is to understand how DNA works within our bodies. DNA was first discovered in the late 1800s by a scientist named Juan Frederick Micher. Micher didn't initially know that he had discovered DNA. All he knew was that he had found some sort of material that came from the nucleus of the cell, and it was not a protein. Because he didn't know what it was called, he called it nuclein. But in actuality, he had discovered DNA. After Micher's discovery of DNA, it began to become more researched by scientists, and by the early 1900s, a researcher named Pueyrredon-Levine discovered that DNA was made up of sugar, a phosphate, and a nitrogen base, which meant that it was a polymer. Even with this discovery, DNA was still not thought to be too simple, or it was thought to be too simple to store genetic information. So the function of DNA was still not understood. In the 1920s and 30s, chromosomes were recognized to carry genetic information, and chromosomes are made up of protein and DNA. The problem was that most scientists believed that proteins were doing the bulk of the work when it came to carrying genetic information because they were much more complex than DNA molecules. It took until 1944 for DNA to be shown to carry genetic material. The way that this was proven was that a scientist named Oswald Avery carried out an experiment in which he destroyed DNA, and when he destroyed the DNA, traits were not being transferred to the offspring, which proved that DNA carried genetic information. The biggest and most important discovery when it comes to DNA cloning was found in 1953 when a group of researchers discovered that DNA was a double helix, which in the future allowed researchers to replicate DNA very accurately through the process of DNA cloning. All right, so next I want to talk about the history of what was considered to be the first DNA cloning. With all the information on DNA that had been discovered in the past 100 years or so, a researcher named Paul Berg was able to clone DNA for the first time. Berg did his experiment in 1972, and when Berg did this, he was trying to answer a simple question in relation to DNA. Can DNA from different biological sources be joined together into one molecule? By the end of his experiment, it was found that DNA can be cut and pasted together to create new types of DNA. Although he found out that different types of DNA could be strung together, he didn't actually clone any of this DNA that he made. So why is this considered the first actual DNA cloning? This is considered the first cloning of DNA because it solved the problem of recombinant DNA. Just for a quick definition on what recombinant DNA is, because it's going to be mentioned a lot, it is a form of artificial DNA which is created by combining the DNA of different organisms together to create a hybrid piece of DNA. The reason that Berg didn't clone this DNA is because he was worried about the safety concerns, because if this strand of DNA happened to get out into public, it could have caused health issues because it could have created some sort of virus. So due to safety concerns, he didn't actually clone the DNA. So the next topic is DNA cloning in bacteria. In 1973, a year after Berg figured out how to create recombinant DNA, two researchers named Stanley Cohen and Herbert Boyer actually cloned this DNA using bacteria. The way that they did this is by adding a piece of recombinant DNA that they created to bacteria, and the bacteria was able to replicate it over and over again. The significance of this was that it proved that DNA could be cloned when put into different organisms, which was a huge breakthrough in the DNA cloning process. All right, so the next topic is safety and regulation. After Stanley Cohen and Herbert Boyer first successfully cloned DNA, scientists led by Paul Berg raised concerns that genetically modified bacteria might escape the lab and cause harm. To address this, researchers, and not the government, organized the Aslamer Conference on Recombinant DNA in California, where they agreed that recombinant DNA research should be continued under a risk based safety system. Experiments were classified by the DNA source, the host organism, and the expected biological risk, and both physical and biological containment measures were established. These guidelines later spread internationally and were recognized by the World Health Organization. All right, so the next topic is the transition to industry. In the late 1970s, DNA cloning became a business because companies realized valuable DNA could be mass copied for profit, and clear safety rules were made the technology acceptable to regulators. A major turning point was when Stanford University and the University of California, San Francisco patented the cloning methods developed by Stanley Cohen and Herbert Boyer, allowing companies to legally license the technology. The first major company built around DNA cloning was Gentech, founded by Boyer and Robert A. Swanson. Its early success producing human insulin showed that cloned DNA could make safe, regulated, large-scale medical products. All right, so the next topic is the effects that PCR had on DNA cloning. PCR, developed in the 1980s by Kerry Mullins, made DNA cloning much faster and more precise than earlier labor-intensive methods that required finding and isolating specific DNA fragments. PCR allows a chosen DNA sequence to be copied millions of times, eliminating DNA scarcity and greatly increasing cloning efficiency. However, because even tiny amounts of DNA were amplified, stricter controls were introduced to reduce the risk of copying the wrong sequence and the sequence getting out and hurting the environment possibly. All right, so the next topic is how DNA cloning works in the modern day. DNA cloning became even quicker and more efficient when computers started being popularized. With the use of computers, researchers can create software to put together different sequences of DNA instead of doing it manually, which makes DNA cloning way more effective and efficient.
So I'm going to be presenting on the concept of DNA cloning, what it does, and all of that. So what the concept of it is that DNA cloning, it makes copies of DNA. It uses a specific gene for scientists if they want to study it or if they want to even use it. And the gene of interest is taken from an organism. It's inserted into a smaller circular piece of DNA. It's called a plasmid. So plasmids are naturally found in bacteria, and they're easy to work with in the labs. So once the gene is inserted, the plasmid is it becomes a recombinant plasmid, which means it contains DNA from two different sources. And so then the recombinant, it's uh, it's placed into a bacterial cell, and so that's where it transforms that bacteria. In that bacteria, the plasmid is copied into the time the cell divides. So that bacteria, will multiply and it'll make many copies of that gene, which is how scientists are able to produce large amounts of DNA quickly and really efficiently. The first step of this is the selection. So this is known as the initiation step, which this is where the double helix unbinds as that DNA is released out. This is important because the strands they need to be separated before the scientists can work with the gene. And so the enzyme with the helix or helicase, it separates those strands, and that's where the replication fork is transformed into. So once that DNA is open, the scientists can see where the specific gene is where they want to copy it, and that's the target gene. And so when that target gene is found, the DNA contains that from that gene that's isolated. The DNA is isolated from that rest of the genetic material. So step two is cutting the DNA. So that's the elongation. This is what it's called. This is where genes of the DNA that run into opposite directions. So you have one strand, which is the leading strand, one strand that's the lagging strand. So what the leading strand does is that it is copied continuously into one smooth process. As you can see up top, it follows the direction of what the replication fork is going, which is where the DNA is getting unzipped. And so the lagging strand is copied into smaller pieces. These are the Okazaki fragments, which is that's the discontinuous side. It's different. The discontinuous goes away from that DNA, getting unwinded, and this continuous means that the leading strand keeps on going, and it just unwinds right there. So the third step is preparing the plasmids. So during the step, the plasmids are cut open, and so that the target gene could be inserted into them. The specific the special enzymes, there are the restriction enzymes. They are used to cut the plasmid at specific locations. This one creates openings in the circular DNA. At the same time, the target DNA is cut used from the same enzyme. So let's make sure the ends of the gene in the plasmid match. And these matching ends, they're called the sticky ends because they are easily attached to each other and they match. So once the DNA and plasmid they have those matching ends, they are prepared for the insertion for the next step. And step 4 is ligation. And so this is where the DNA ligase starts to join those fragments. DNA ligase is the main part of this whole step. It works just like glue. It seals the DNA fragments together. The DNA ligase it connects the matching ends of the gene and the plasmid then forms one complete piece of DNA. And so once those are joined, that's where we get the new DNA called recombinant DNA because it contains DNA from 2 different sources. And so the recombinant DNA is now in a form of a plasmid that carries that inserted gene. And so the recombinant plasmids are what will later be placed into the bacteria so that they will be copied. And this is important because this is where the final DNA molecule is needed for cloning. So then begin to step 5, which is transformation. So this is where the plasmids enter the bacteria. And in this, to help the bacteria absorb that plasmids, the scientists use a process called heat shock. So this involves briefly exposing the cells to a higher temperature, which is what it says in the name, the heat shock. So it makes tiny openings in the bacterial cell membrane, and it allows DNA to enter that. So as a result, some of the bacterial cells taken into that recombinant DNA. But not all cells are going to successfully take up the plasmid, which is why this is going to be focused on selecting the correct bacteria. And so we get into the selection, which is how the antibiotic resistance is how the bacteria is placed on a plate with many antibiotics. This is only the cell that is took in the plasmid that could survive, because this is where the scientists could easily identify the correct plasmid from the incorrect ones, or the more modified cells from the unmodified cells. But another method that they use for this is the blue-white screening. This is how we see the gene expression get put into play. So we can see that the bacteria with the correct plasmid, they'd be white, while the ones that are not, they're blue. But at the same time, the surviving bacterium, they'll grow and it'll divide and they'll become a colony. which is when the colony contains the same plasmid, the same copy gene. So for the production of the copies, this is where the bacteria will instantly split. And the bacteria reproduces very quickly through binary fission, which is where one cell splits into two. So every time bacteria it divides, it also copies a plasmid inside of it. And since each plasmid contains the target gene, this means that every division creates more copies of the DNA. So as bacteria continues to multiply, the number of the plasmids will increase. And in a short amount of time, uh, scientists will produce millions of copies of the same gene. So this makes DNA cloning a powerful tool for research, for medicine, and for biotechnology.
DNA cloning is essential for safe genetic research. It allows scientists to create multiple copies of genes for study without using the organic samples or harming others. We use cloning to analyze three main areas, and that includes the gene function, mutation of the genes, and how the genes interact. By cloning and studying DNA in these controlled lab settings, scientists are able to take away all the other factors so we can actually get accurate research and safe research on these topics. The next big topic of relevance for DNA cloning is genetic engineering. Now, genetic engineering is where doctors insert clones of the DNA into any organism they need to. They do this into faulty organisms where they have faulty genes and they insert other genes to help counteract the faults. They can do this by introducing, removing, or modifying certain genes. The methods of genetic engineering and inserting these genes include electroporation and something called gene guns, which is how they actually put the new modified genes into the old organism. The first area of importance in DNA cloning is medicine. DNA cloning has not only led to a lot of advancements in medicine, but it also helps us not rely on scarce resources. So, how they do this is they clone DNA, human DNA, and mix it with bacteria. They use this to create medicines. This includes insulin, growth hormone, and TPA. And they can do this all without relying on scarce natural resources and instead relying on cloned DNA. The next area of importance is gene therapy. So DNA cloning is really important because it allows us to do gene therapy. Now, gene therapy is where we modify or create helper DNA or genes that train the body and combat faulty genes by training the body to create more of those helper genes to either limit the damages of those faulty genes or completely cure it. This helps cure and support diseases such as cystic fibrosis, uh, muscle dystrophy, and immune system defects. These diseases had a lot of struggle getting cured and did not have a lot of advancements in the medical field with these diseases until DNA cloning and gene therapy.
Oh, there we go.
The final and maybe most important part of DNA cloning is the advancements and effects it has on our agriculture. DNA cloning is used to clone useful genes to either boost nutrients or for pest control in our agriculture. Examples of this can include the Bt toxin gene, which is cloned in the corn to help protect it from pests and that corn getting ruined. And also, golden rice has a very similar pest control gene. And scientists also clone nutrients to boost the vitamin A to make that even healthier for us humans to consume. One very recent application of DNA cloning was last year in 2025, how DNA cloning helped improve cancer vaccines and treatments for cancer. They did this by cloning each patient's specific DNA to create hyper-personalized treatments and vaccines for those types of cancers and for that person's treatment as their own genetic field. They used these clone genes and these personalized genes to train the patient's immune system to fight these cancer cells and to help stop the spread of the cancer. This improved survival rates on a lot of very hard to treat cancer types, and a lot of them that weren't very popular and hadn't had a lot of research done into them. In summary, DNA cloning is very useful because it helps not just the medical field in curing diseases and helping gene therapy, as well as creating a lot of types of medicine, but it also helps our agriculture and boosts those organisms that we eat and live off of. And here are our references. Thank you so much for your time. Thank you for watching.